Please, the floor is yours.
Well, good morning, everybody. It's a pleasure to be here. It's nice that the weather, it looks as though it may be clearing up, which is good. What I want to do today is to give you pretty much an overview of bacterial methylation and talk about some of the interesting findings that have come out for this. I think everybody knows about epigenetics in humans and a lot of interest in that at the moment. Much less is known or talked about as far as bacteria are concerned, and yet bacteria do a lot of methylation. So I'll give you an introduction to that, talk about what we know, and then talk about where we might go with that eventually. Is this supposed to work? Yes, okay. So basically in bacteria, there are three types of methylation. There's 5-methyl-C, which is the type of methylation you find in eukaryotic cells, in humans and so on. There's M6-methyl-A, which you find in some eukaryotes, but to a much lesser point. And then there's M4-methyl-C, which is only found in bacteria and archaea. Now you might ask, why would you want to study methylation in bacteria other than out of intellectual curiosity and it's fun? Well, they're part of restriction modification systems, and because of my long-time interest in restriction enzymes and restriction systems, we discovered in my lab at Cold Spring Harbor 75 out of the first 100 restriction enzymes that were discovered. And so I've had a long-time interest in the biology in this area. The genes for methyltransferases are very easily found by bioinformatics, and so that's kind of nice. And they serve as good markers for restriction modification systems. In some cases, we know they have regulatory functions. I'll mention that a little later. They support epigenetic phenomena, and this is something that we know much less about. I'm not going to go into detail in that, but there are several groups around the world now who have been studying this. We know that, for instance, in the case of Neisseria infections, Neisseria meningitis in particular, that there is a type 3 restriction modification system there that is responsible for switching methylation patterns and extending the host range of Neisseria. They're also very good examples of proteins that recognize DNA sequence. You know, everybody's interested in transcription factors and other things that can recognize DNA sequences. Methyltransferases do it in some interesting ways. They also have this interesting property called base flipping, which was first discovered in restriction systems in the HHA1 methylase. And this is when the protein interacts with DNA, wants to methylate the base, and it flips the base right out of the DNA helix, as shown here. I was lucky. We just discovered this about 6 weeks before I had to go to Stockholm to give my Nobel lecture. And since I'd not actually worked on splicing for many years prior to that, I chose to talk about this. And of course, if you make a discovery, there's no better place to talk about it than Stockholm. So this is very interesting. It turned out after this was discovered that pretty much every protein that does chemistry on DNA uses this same mechanism. It flips out the base in order to then go ahead and do the methylation or the chemistry. From my perspective, one of the really interesting features, there are many more known now that appear necessary for the biology that we know about. And so that means that there is stuff going on biologically with these methylases that is not well understood. And to me, that smells of a discovery waiting to be made. I'm kind of hoping I will make it, but if one of you were to make it, that would be fine too. Now, I want to talk about the restriction systems in particular, because this was how I got interested in this in the first place, and they actually turn out to be fascinating from many points of view. So there are three basic kinds of restriction systems that use methylation to protect. First of these are the type 1 systems. These were the ones originally discovered by Werner Aber back in 1965 to 68 or so. They're composed of 3 genes. There is an R gene that is responsible for restriction, shown here in orange. In blue is the methylase gene, which is responsible for the methylation. But the key gene is the S gene. This is the gene that provides the specificity. So the specificity gene makes a protein, usually about 400 amino acids long or so. It binds to DNA, and then if it combines with the methylase, 2 copies of a methylase gene, it will methylate DNA. That's all that's required for methylation. But if you want to restrict unmethylated DNA, then you also need to include the R gene, 2 copies of the R gene. Turns out recognition sequence determination there was always very difficult, and that's because the restriction enzyme cuts randomly the DNA. So you don't get nice discrete fragments. You can't use those fragments to work out what is being recognized. You have to do it by studying methylation and ask what sequences are methylated in order to discover the recognition sequence. That turned out to be quite difficult until Pacific Biosciences sequencing came along, and then all of a sudden it became very easy. The type 2 enzymes, these are composed of 2 separate genes that operate completely independently of one another. There is an R gene that is responsible for restricting DNA, and they have the advantage that they're specific. They cut DNA at very specific points. And these were the ones that became the workhorses of biotechnology. This was the basis upon which New England Biolabs was formed. And they're fascinating enzymes. They're really interesting, many different subtypes of these enzymes, but they all have this property of cleaving specifically. They are accompanied by a methylase gene that operates totally independently of the restriction enzyme gene. It methylates this same recognition sequence and sometimes recognizes other sequences too. So it's not as specific in many cases as the restriction enzyme, but in lots of cases they have exactly matching specificities. We don't really know why that should be so. If you think about it, there's no reason why you couldn't have a methylase that methylated, say, all the A residues in a genome, and a restriction enzyme that just recognized GAATTC. The methylase could still protect against the restriction enzyme. So you might think it's a little strange that they would have matching specificities at all. There, the recognition sequence for the restriction enzyme was always very easy to find because you can just look and see where the cut is, and you know that the recognition sequence is going to be close by, very often right in where the cut is. The methylase, again, was a problem. Discovering and working out methylase recognition sequences prior to about 8 or 9 years ago was very difficult. The sort of thing students didn't want to do. You would ask a student to do it, and they'd go and talk to other people who'd been doing it, and then come back and ask for another project. So not so easy. The type 3 enzymes are very interesting in many ways. They have been known for a long time. The methylase is quite hard to find the recognition sequence for exactly the same reasons that the other are. The R enzyme was a little bit easier because the R is specific, but the problem is the R enzyme rarely gives complete cleavage. It's very difficult, actually, in order to get good cleavage with the type 3 enzymes. You usually have to work pretty hard on the biochemical conditions to do that. The recognition specificity here is all in the M gene. So no R gene, the R gene has no specificity that we're aware of. The M gene provides the specificity. To methylate, you just need the M gene in most cases, although we've recently come across cases where that's not true, where you need both the R and the M gene just in order to get methylation. And if you want to get restriction, then you need both genes because you need the M to provide the specificity. Now, in 2009, there was a paper by Ede et al. From Pacific Biosciences pointing out that when you do PacBio sequencing, you can also start to see where the methylation sites are. This was followed by a couple of papers listed here that go into more detail about how to do that. The way in which it works is that when the polymerase is copying a template, here the polymerase is actually tethered on a microbead. When it's trying to copy a base that is methylated, it slows down. The polymerase has to spend some time figuring out what base has to go in opposite a methylated base. And because PacBio, the machine, is a single molecule machine, it measures the time of addition, and so you can get a signal here, which is, they call it the interpulse duration. It's the time from the addition of one base to the next. And when you start to look at these IPDs and plot them out, in the case here, GATC, when it comes to the C, there is a big gap in time before that base goes in, and so you know there's a modified C on the other strand. Turns out that with this method, you can get very good data for N6-methyl-A, you get pretty good data for N4-methyl-C, very variable data or no data at all for 5-methyl-C. So this is not the method of choice for 5-methyl-C, although there have been some modifications introduced that will modify 5-methyl-C and put a bulky group on it, and you get slightly better signals, but you still can't guarantee it. And so this is not the method of choice to look at 5-methyl-C. Now, we got involved in this. I got excited when I read these papers and talked to the people at PacBio, because what we had at the time were a very large number of clones of restriction systems, because these were our production strains, in which we had both the restriction enzyme and the methylase, and we had strains of E. coli with no other methylation on, so that the only methylation was due to a known gene. And so we said, why don't we send you some of these? We can check whether your method really works, whether the software is up to really telling us what the recognition sequence is. And they were delighted, I have to say. Some of the best collaborations I've ever had have been with PacBio. They're very academic friendly. They're interested in the underlying intellectual aspects of this. So we sent them these 3 clones. At the top is the DAM gene from E. coli, does GATC, methylates the A residue at M6A, and you can see you get a very nice signal here. We also had one from an environmental sample that did 4-methyl-C. When we did that, we got a nice signal, but there's a little bit of background, you can see. And then we took the gene from Staph aureus that recognizes GATC and makes 5-methyl-C, and here you see the problem with 5-methyl-C. You get a very broad signal. The real signal is shown in brown, the blue is sort of background. And this actually is quite a good signal for an M5C with PacBio. So we then went on and said, well, why don't we send you some things where we know the sequence that's being methylated, but we don't know which base is being methylated within that sequence. We looked at this one, AATT. We didn't know whether it was the first or the second A that was being methylated. Got a very nice signal corresponding to the second A. Down here, we actually didn't know whether it was N6A or N4-methyl-C, and turned out to be N6A, and again, we got a nice signal here. So we actually looked at about 20 where we knew the answer, and we started looking at things where we didn't know the answer, all type 2 systems. So most of our clones were type 2, and we got some very nice results from this. And then we thought, well, can we look at type 1 and type 3 systems and get equally good results? And so we sent them some of those. This is a type 1 system from an A strain of E. coli. Has a nice recognition sequence. We knew what it was, and PacBio gave it immediately. We also talked about type 3. Can we do type 3? This is from a Burkholderia species, recognizes CACAG, and again, gave us a very nice signal. This was the sequence from a Bacillus cereus strain. We didn't know what the recognition sequence was ahead of time, but when we looked at just a few sites on a plasmid, there are just 6 sites on this plasmid, got a very nice consensus sequence, CGAAG, with the second A residue being methylated. And so at this point, we put out a paper and described what we've been doing. We then went on and thought, well, these were all done on cloned genes, I should say. So in every case that I've talked about so far, we cloned the methylase gene into an E. coli strain, did no other methylation. And then we thought, well, maybe we don't need to do the cloning. Maybe we can look at whole genome sequences and do it. And so we took a series of 6 bacteria. We'd already got the sequence of the strains, so we knew how many methylase genes were in each strain. In some cases, we could guess what the methylation pattern was going to be. In others, we had no idea. One of the first ones we did was Chromohalobacter selexigens, had 3 methylase genes in it. One up at the top here was clearly part of a type 1 system. It has the R, the M, and the S gene. And these were 2 solitary methylases. We didn't know what any of them recognized. So we did a whole genome analysis and found that there was a methylase recognizing RGATCY, another one recognizing this more complicated sequence. Now, these split sequences are absolutely typical of type 1 systems, and so we're pretty certain it was due to this. This must be due to one of these, but we didn't know which one. And so we cloned 3 separate systems, checked that everything was okay. Sure enough, this was the type 1 system corresponding to this. And it turned out that this one corresponded to this methylase, M.CSA-A1. And this one recognized a relatively non-specific sequence, M6A followed by anything other than A. That's what B means in this case. This turned out to be on a prophage. Prophages, it turns out, very often have methylases on them. Presumably, when they pop out of the genome, they turn on the methylase and then protect themselves against would-be restriction systems in any bacteria they're going to infect. And that turns out to be much more common than anyone had expected. These things are not usually expressed in the prophage. So when you do the whole genome sequence, you don't really detect the expression of this particular one. So we did a total of 6 strains. These are shown up here. They had a variety of type 1, type 2, or type 3 systems. Total of 27 methyltransferases between them, 13 of which were new specificities that had never been seen before. And again, in each case, to confirm that our predictions were correct, we cloned out the genes and tested them all individually, because we were anticipating doing a large amount of this, and we wanted to be sure that we were going to be able to interpret everything properly. Now, this was the first strain that turned out to be really interesting. And again, it's a case, we got the PacBio sequence, Pacific Biosciences did the sequence in collaboration with a group from Mount Sinai Hospital. This is the strain that caused the enterohemolytic problems in Germany. There was just some major, I think 50 people, 60 people died. It was a food contaminant. Good reason not to eat organic. You know, if you want to pick up a bacterial acquired infection, pick organic food. 10-fold increase in foodborne infections that way. Anyway, we saw that there were a large number of systems here. And it's interesting, the reason that this has so many systems is because it had gotten rid of this MCR, BC, MCRA, MRR systems. These are restriction systems called type 4 that specialize in cutting DNA that is methylated. Okay? So in E. coli, there is a little island that has these genes on them. The recognition sequences for these particular type 4 enzymes do not match those of the normal E. coli. And what had happened in this particular strain, this entire island of methyl-dependent systems had disappeared. And so that then allows strains to pick up extraneous DNA very easily. And that's what happened down here. Just want to talk about a couple of instances of specific methylases here that turned out to be interesting. The first was this one, Echo G1 and Echo G2. They're almost identical genes. They're on prophages within this E. coli strain. They recognize A. So the recognition sequence here is A. And in vitro, you can get about 96% methylation of all the A residues in any DNA that you treat with this. These are not expressed in the host strain. We only found these because we cloned out all the genes in order to be, we thought it was important to check in this case, make sure everything was okay. This is what it looks like if you look at the plasmid. These are the two strands that are methylated, two complementary strands. When you look at the consensus sequence, this is what it looks like. It's got a slight preference for a C in front of the A. And it's very useful if you want to pre-methylate DNA for transformation. Biolabs sells that as a product now. That's the only advertisement I'm going to give you, though. Okay. Now, there was another one that turned out to be really interesting, and this one is called M.EchoG9. It turns out it's A-specific, also single-strand specific. It just methylates one strand of the DNA. Again, here we look, this is the outer strand, this is the inner strand of this particular plasmid. And every time there is a blip on it, this is where there's a methyl group. Now, this only does maybe 20-30% methylation and only does plasmids. In Burkholderia, there's another one here. Again, just methylates one strand of the DNA. And so we were curious as to how this could happen, even why it might happen, but how does it happen? And our immediate thought was, well, maybe what is going on here is that this particular methylase is not being used as a methylase. It's not the methylation that's important. It's actually being used as a helicase to open up the DNA. And it's taking advantage of the fact that when the methylase binds to DNA, it flips out the base. And that means that these two bases have lost their stacking interaction. So half the stacking interactions have gone. And so this could then easily allow the thing to go on. And so we speculated that it might be part of the replication machinery. We have now done some experiments to test that hypothesis. But the first thing that struck us was that if it is a part of the replication machinery and is only doing one strand, if we were to switch the orientation of the origin of replication on the plasmid, then perhaps the other strand will get methylated. And sure enough, that's exactly what happens. So when the origin is facing in an anti-clockwise direction, then the inner circle gets methylated. When it's in the other direction, it's the outer strand that gets methylated. We now know if you try to purify this methylase, the DNA polymerase 1, which is the replicative replicative polymerase for this particular plasmid, that it comes along and the two purify together. We have some other experiments we're just finishing up at the moment that will clarify and solidify this, and we're about to publish this. Now, thanks to PacBio, there's been just a tremendous increase in the discovery of methylases. So the first methylase was characterized in 1969. This was one of the E. coli methylases. And then for a long, long time, there were almost no recognition sequences determined. And then along came PacBio, and now you can see this is the rate of discovery of new methyltransferases just because of PacBio sequencing. This means that anybody who ever does any PacBio sequencing should really look at the methylation analysis. Unfortunately, a lot of people don't do that, but it really leads to the discovery of a lot of new information. And if ever you want to work on a strain and you want to transform it, it's very nice to know what are the recognition sequences of the restriction systems that are going to stop you getting DNA into that strain. We've also found along the way that we've come across a lot of very novel systems. First thing we came across, here's one, a normal type 3 enzyme recognizes CACM6AG. And we've now found quite a lot that use N4C. So they use cytosine as the protection. We probably have about 20 of these now that we know about. Also came across some interesting novel type 1 systems. Normally, as I told you, there are 3 genes, R, M, and S. In the case of this particular system from Pseudomonas alkaligenes, the recognition sequence is CCC N6RTTGY. These three C's, there's no A on either strand, and so they can't use A to protect, and so they use N4C to protect. And when they do that, there are two methylase genes, one of which does the N6A, and the other one does N4C. And we have, this is all published stuff, we know about it. We now know there are a lot of systems like this, and so this is a very nice bioinformatic marker. If you see a type 1 system and it's got two M genes next to it, you know that it's going to be using N4C to methylate. I want to talk a little bit now about the type 1 specificity subunits because they turn out to be interesting. Typically, they're about 400 amino acids long. They have two regions in them, varying from sort of 100 to 150 amino acids, that recognize the two halves of the recognition sequence. So this target recognition domain here recognizes the 5' most sequence, the one at the C terminus of the protein, again similar length, and it recognizes the second half, the 3' half of the recognition sequence. There's a lot of variation here. So we see lots of variation in terms of the sequences present here, sequences present here, and the number of N's that are present. Some of these S subunits are quite complicated. They have more than 2 TRDs, not a lot of them, but there are some. They may have 3 or they may have 4 TRDs. And at the moment, it's not easy to decide which 2 are going to be used and whether or not, in some cases, more possibilities are there. So maybe you use TRD 1 and 2, or TRD 1 and 3, or 2 and 4, and so on. We're not all that clear on the rules for doing that yet. The actual TRD sequence can be anything from 2 to 5 bases long. If it's 5 bases long, that suggests that you could recombine these and actually make a recognition sequence 10 bases, 5 on one side, 5 on the other. We know that it's easily possible to do this both in vivo, this happens frequently, and in vitro we know how to do that too. The submotifs can also vary in sequence. B means anything other than A, K is A or C, and so on. V is something, not T, anything other than T. R is purine, Y is pyrimidine, and so on. So the submotif sequences vary quite a lot, and I think it doesn't take much calculation to figure out that the total number of possibilities here is absolutely enormous. And I can guarantee that at the moment, if we sequence a new bacterium, it's not been sequenced before, it's got a type 1 system in it, there's a better than 50% chance, maybe 70% chance it's going to be a new specificity that's never been seen before. The number of N's separating the submotifs varies from 4 to 9. Those are the ones that we've seen so far. It's possible that there might be shorter or longer ones, but these, among the several thousand that we've seen so far, these are the variations that we see. Now, we've just, a computer programmer who works for me, János Pasfai, who's a Hungarian computer scientist who's worked with me for many years now, has been analyzing these so that we can try to predict what TRDs are recognizing what sequences. We've now been able to make predictions for about 4,000 different TRDs and which is recognizing which half of the sequence. That work will be written up in the next 4 to 6 weeks. So it's turning out that it's very interesting in the sense that if you determine, say, you've got ACG in one half, there are more than one way of doing that. You can find TRDs that don't look anything like one another that recognize ACG. And so there may be, for any given half motif, there may be 3 or 4 completely different protein sequences that can do it. So it's not just as though there's one protein sequence that always does the same recognition sequence. So what have we learned so far? Well, we've learned that there are an awful lot of solitary methyltransferases that don't appear to have restriction genes associated with them in both bacteria and archaea. And it's very tempting to think that these are probably involved in epigenetic phenomena, but we don't know for sure. There's much more variability in recognition sequence than was expected. Huge variations, both among the type 1s, the type 2s, and the type 3s. The type 2 methylases often recognize exactly the same sequence as the restriction enzyme. So maybe GAA-TTC, both the methylase restriction, recognize exactly the same thing. But there are also some occasions, and maybe a little more than we thought about a year ago, where the methylase recognizes slightly more sequences. It's slightly degenerate. And if you think about it, let's say you've got a restriction enzyme that recognizes GCC-GGC. A methylase that recognized GGCC would always protect against that restriction enzyme, but it's degenerate. It recognizes other sites too. And so that makes it a little tricky. You have to be careful about interpreting restriction sites, recognition sequences on the basis of the methylases. But it also raises an interesting question, and that is, why would they be the same? Why should, at any given time, the methylase and the restriction enzyme recognize exactly the same sequence? And this was something that we'd sort of thought about a little bit, but no one really had paid great attention to it until I gave a talk one time from a hotel room in Japan to a conference in Novosibirsk in Russia. And I gave a methylase talk, and at the end of it, one of the people in the audience said, is it possible that what's being selected is not restriction, the ability to stop phages getting in, but could it be it's the methylase that is actually being selected, and that the restriction enzyme is just there to keep the methylase honest and make sure it recognizes the same sequence? I'd never been asked that question before. I've never been asked it since. I tried to find out who asked the question because I thought it was actually quite an interesting possibility that it was really the methylase that was being selected, but no one fessed up to asking the question when the organizers were sort of trying to do this. It was several weeks after the talk, I must say. But I'm still trying to find this person. If they're in the audience, let me know. So the other thing I think is that in prophages, when bacteriophages infect a cell and then get embedded in the genome, they turn everything off. Usually the methylase gets turned off too. There are an awful lot more prophages that carry methylases than one might have expected, and presumably this is to defend when they're going to enter a new genome. So I want to talk just a little bit about some of these common orphan methyltransferases, the things that don't come with a restriction counterpart. So the GATC methylase in E. coli is pretty well known. This is involved in replication. It's also involved in DNA repair and also can affect transcription. So sometimes a promoter can be methylated at GATC and turned off. In Colobacter, there is a methylase that recognizes GANTC, and there are a large number of related organisms that have this same methylase in them. And we know that this is involved in the control of the cell cycle. It can decide whether cell is going to start replicating or whether it's not. This has been studied pretty extensively by a number of groups now. In the Burkholderia and related organisms, Pandorea, Cupriovidis, and so on, there is this methylase that recognizes GT, WW, W is just A or T, AC. It is present in almost every Burkholderia strain. We have no idea what it does. We would like to find out where at Biolabs we can't work with pathogens because the employees are a little concerned about that, at least the non-research employees. And so I'm trying to find a lab who work on Burkholderia to explore this a little further because I'm betting it's doing something interesting here. In the Clostridiales, particularly in C. difficile, which is a really common hospital-acquired infection these days, which has problems, there is a methylase that does CA5, methylates the last A base. We've been working with a group at Mount Sinai Hospital on this, And we have sequences for literally hundreds of Clostridial strains. They all have this CA5 methylase in. And in C. difficile, if you knock out the gene for this methylase, C. difficile can no longer sporulate. And so that looks as though it could be a very interesting target for an antibiotic if one could find one specific for this, because one of the problems with C. difficile is that often it's antibiotic resistant, but even so, it's a persistent infection, and presumably this is because it sporulates, sits there, the antibiotics can't get at it, and then it crops up later when you stop the antibiotic treatment. And so this might be very, very useful, because as many of you probably know, the optimal treatment for C. difficile infection is a fecal transplant. Now, these are done a little more felicitously than they used to be, so they don't pour shit down your throat anymore, but they actually put it into tablets, and you can take the tablets, and away you go. And finally, I would point out this thing in Salmonella. So CAGAG is a methylase that is part of a type 3 restriction system that is present in every Salmonella. We don't know why it's there. We don't know why there should be a type 3 system. These are not particularly good restriction systems usually. And so for me, this is one of the best examples we've come across so far, whereby maybe it's the methylase that is being selected for reasons that we don't understand, and the restriction enzyme is just there to keep it honest and make sure it keeps recognizing this sequence. I want to make a plug for PacBio sequencing. It's really, we've had such good success with it. Methylase recognition sequence are easy. Functional annotation of methylase genes becomes straightforward. And so just from getting the genome sequence, you get the methylation profiles and annotation. Closing bacterial genome sequences has become almost trivial. It's very rare that after one run with a PacBio sequence, you can't completely close the genome, which is not true of any other method at the moment. And the accuracy of sequence is enhanced by going through, even though any individual PacBio run, just one run on a molecule, is not terribly accurate, the errors are random, and you go through every sequence many, many times, and so you end up with very accurate sequence as a result of doing this. Now, I just want to finish by telling you about something that we've been doing very recently, and this is an alternative way of looking at M5C methylation. So I think if any of you have ever done C5 methylation studies and try to work out which bases are methylated, probably have used bisulfite sequencing. That is probably the technique that I dislike most in the lab. I personally did it many, many years ago. It's horrible, it's awful, doesn't give terribly good results. And so we've been thinking for a long time how we might do a better job. Well, a few years ago, we discovered this enzyme called MSPJ1. It is a methyl-specific enzyme. It recognizes 5-methyl-C, followed 2 residues later by a purine. So the recognition sequence is M5C-NNR. Now, if you think about a typical C5 methylase, they're usually palindromic, and so this enzyme cuts 12 and 16 down beyond the sequence. If it's palindromic, it does the same thing on the other side. And so you end up with a fragment that is anywhere from 30 to 32 base pairs long that contains the recognition sequence right in the middle. For this, you can take a genome, shown in lane 3 here. You get a nice genome, whole genome, cut it with MSPJ1, and you get this nice 32 base pair long fragment that has all of the M5C recognition sequences in it. And so all you then have to do is to sequence all of these 32 base pair fragments. We've been doing that with Ion Torrent, but we've also done it with Illumina and with PacBio. And then by doing a little bioinformatics analysis afterwards, you can work out what are the recognition sequences. And we found this to be very good. If the C5 methylase is expressed, then you're in great shape. If it's not normally expressed, you may have to clone it first in order to be able to do this. But it's made it very easy now to determine M5C recognition sequences. Again, we're just actually trying to polish the bioinformatics to do it a little better than we have in the past, and we will publish this very soon also. And I mean, it's really going to change one's ability to get C5 methylation recognition sequences. Now, I should say, when we first found this enzyme, we immediately thought of using eukaryotes and looking at eukaryotic CpG methylation. It works for that, but it doesn't work as well as you might like. And the reason is because when CpG gets methylated in most eukaryotes, there is a phenomenon called methylation spreading that can often take place. And so nearby C residues can also get methylated. And also the methylation in eukaryotes is often not complete at CG. And so you get partial information, and it's difficult to get quantitative data out of this. But for bacterial methylases, it works just fine. We now have sequences for about 40 C5 methylases that we didn't have before, and we've been working with a lot of groups to extend this. Want to acknowledge people who've been involved in this. So in my group, Ian Murray, Brian Anton, Alexei Fomenkov, Janos Poshfai, Rick Morgan is a fellow investigator at New England Biolabs, a fellow scientist, and he's been doing a lot of this stuff too. At the Sanger Center, they've been sending us a lot of their PacBio data for analysis. At the Broad Institute, they used to send us data. They've not been sending it recently. PacBio, principally Tyson Clark, Jonas Korlak, have been just fantastic collaborators. They've been just excellent. At Harvard, Laurie Comstock, Sloan Kettering, Gang Fang, he's the person who was responsible for writing some of the PacBio software originally. He is the person we've been collaborating with on the C. difficile data, and that has been submitted for publication at Nature Microbiology. At the moment, it's in revision. In Malaysia, there are two groups that we've worked with pretty extensively, and then a lot of others. There's something now, I think about 150 groups have sent me PacBio data so that we could do analysis, and then we make it available through Rebase, this database that I run. I would also ask you to think about joining the Nobel Laureate campaign to support GMOs. This website, supportprecisionagriculture.org, has a place where you can sign up and say, well, I'm with you. We have 137 Nobel laureates already signed on to this campaign, and we're trying to convince the European, in particular, Greenpeace, to just admit this is something they got wrong, and let them get on and do the stuff they can do well, and not go telling the developing countries that they shouldn't be growing GMOs. The developing countries need them. If they don't get them, there are going to be a lot of dead people down there. And when people get hungry, as we've already discovered, they like to head north. And if Europe thinks it has an immigration problem now, just wait 10 years and see what's going to happen. The population of Africa is exploding. By the end of this century, it's going to be the most populated continent. I thank you all for your attention.